Okay, so let's get a few things straight. Cellular respiration is the process by where organisms use glucose, the chemical energy in glucose, in the presence of oxygen to break it down into products that have less energy in order to release energy to make something called ATP. That's the purpose of cellular respiration. We need organic molecules that start off as glucose. We break them down into their pieces, and energy is released in the form of electrons. And we take those electrons and we're going to make energy from them. Okay? There are three really important molecules that will help you understand the process. All right? Glucose is one of them. All right? Let's move forward here. This molecule, glucose, this is where it all starts. Okay? Don't worry about these reactions right now. Glucose is the molecule that is harnessed and used for these processes. Now, as you can see here, fructose is also a molecule that is important in the process, and the fructose can enter the molecule at any time. All right. Now, think big picture again. It's all about the hydrogens on glucose. And what do the hydrogens have that we need, that we can harness? They have electrons. All right? So the cell is going to harness electrons in the form of hydrogens in the process of breaking down glucose and taking these electrons to where we can make ATP. In some cases, we are lucky enough to be able to make ATP directly from these reactions, but it is not the norm. Okay? It's not the common thing to do. Sometimes we can make ATP directly from the organic molecules, but most times we can only harness electrons first. Okay? So, cellular respiration is a process whereby we're using the chemical energy to produce ATP in both the cytosol and the mitochondria of plant and animal eukaryotic cells. Prokaryotic organisms only do glycolysis. They do not have organelles to do this process here. The mitochondria is a energy-making machine of which only eukaryotic organisms have. Okay. Now, there are two really important molecules that we should probably focus on. One of them is adenosine triphosphate. Okay? This molecule here is called ribose, the green. It's attached to something called a nitrogenous base called adenine. All right? And these two molecules are attached to three phosphate groups. The point of cellular respiration is to take ADP, adenosine diphosphate, with only two phosphates, and to force this third phosphate on to make the molecule more unstable, to make it more reactive, so that we can then use the instability of this to drive other reactions. Okay? And if we look over here at the ATP cycle, ATP is made into ADP and it releases energy for cellular work and making things, an anabolic chemical synthesis to make. When it becomes ADP, we then have to use energy in the form of sunlight for plants or food for animals in order to force this third phosphate back on to make ATP again so we can use it. And this energy is going to come from glucose in some form. Okay? You're going to eat lots of stuff in your life, all right? But all of it the energy part of it is going to be made into the glucose molecule by your body in order to run through these reactions. So, ATP, the chemical energy molecule of the cell. The point of all of these processes to make ATP from ADP using the electrons from glucose. Our next important molecule is nicotinamide adenosine dinucleotide hydrogen, NADH, which is an electron carrier. Remember, we cannot make as much ATP as we want unless we first harness the electrons from organic molecules and take those electrons and put them to work to make ATP. So, throughout these processes, you will see that in these reactions, many times we will make NADH from NAD+ by taking 2 electrons from organic molecules in the form of 1 hydrogen and 1 free electron. NADH is an electron carrier. All right? This whole process is about making ATP from organic molecules by using electrons' energy. Okay? Now, this is just a graph that tells us that electrons will move or jump from atoms or molecules of lesser electronegativity to greater electronegativity. That is why NaCl makes a ionic bond. Because chlorine is taking an electron from sodium, making it positive. Chlorine becomes negative, and the molecule forms a bond due to unlike charges. Okay? So, the cell will use atoms like this to get electrons to move from NADH onto these molecules. All right. So, big picture. All right. We talked a lot about glycolysis. All right. And the 10 reactions and why they're important. Okay. We produce 2 net ATP, 4 total ATP, 2 net NADH in this process using glucose to pyruvate. We produce 2 pyruvate. All right. Happens in the cytosol. It's the first process. All right. Prokaryotes only do this. Any NADH produced in any part of the cell is going to go to a specific place in the mitochondria to get used. Remember, NADH carries 2 electrons. Anytime NADH is produced, it's going to get transported into the mitochondria to get used. That includes FADH too. All right. Some terms that are important: metabolism, anabolism, catabolism, endergonic, endothermic, exergonic, exothermic. 10% rule: glucose, ATP, ADP, adenosine triphosphate, adenosine diphosphate. So, getting a little bit more specific, okay? We focus first on glycolysis, the process of making 2 pyruvate molecules, 2 3-carbon pyruvate molecules from glucose. All right. Now, we talked a lot about the specific reactions here, okay? So in your notes, you have what is happening in each reaction and why each reaction is important. Well, let's focus on the big picture here. The cell is going to use 2 ATP, 1, 2, in order to get 1, 2 ATP back per pyruvate. But remember, how many pyruvate are we making out of this process? There's 3 carbons here. We're missing 3, right? So there's 2 pyruvate. So we're making 1, 2 for 2 pyruvate. So we should be making 2 more. So if we're making 4 ATP here, from step 6 on, it's the energy payoff phase. All right, we make 4 ATP, but we've used 2, right? So we're netting 2 ATP because we use 2, but we make 4. So when all is said and done, we've really only made 2 to use. All right. We also produce 1 NADH electron carrier per pyruvate. Remember, from step 6 on, we're creating 2 of each. Right? So in glycolysis, this whole process occurs in the cytosol, is to take glucose, break it down into pyruvate, harness some electrons, and make some chemical energy in the form of ATP. All right? Now you also need to be familiar with each reaction and why it's important and why it's happening and what's going on. All right? But right now I want you to focus on the big picture. All right? Because I think by now you'd probably tell me this reaction is important because it makes this molecule more unstable and it keeps it inside the cell, right? You could tell me that this reaction is done to make the molecule more symmetrical, because we're going to break it later. And you could probably tell me in this reaction that we're using a phosphate from ATP to make this molecule even more symmetrical, or even more unstable. And you could probably tell me that this reaction splits the molecule into two, so we can harness more energy from each as a smaller molecule instead of one bigger molecule. You could probably tell me that step 5 is to take this molecule and make it into an identical molecule called glyceraldehyde 3-phosphate, so that we don't have to run two completely different catabolic processes that would waste energy. You could probably tell me that this reaction harnesses 2 electrons from the molecule. That's really important. And we use a phosphate to put onto the molecule without using ATP. And the fact that we're not using ATP to put a phosphate on is really awesome and easy and great for the cell. You could probably tell me that this reaction here is used to harness chemical energy in the form of ATP from an organic molecule. That this reaction, number 8, is designed to change the molecule so we can later reap energy from it. That this reaction is also designed to change the molecule so we can later get energy from it. And that step 6 reaction is to take a phosphate off to store it as chemical energy in ATP. And then overall, glycolysis produces 2-pyruvate, which will go to the pre-TCA and TCA cycle in the mitochondria. Alright, you could probably tell me that the pre-TCA reaction of pyruvate made into acetyl-CoA, which acetyl-CoA is going to go into the TCA. Remember, acetyl-CoA goes into the TCA cycle, and this cycle cannot run without acetyl-CoA. So if acetyl-CoA wasn't there, this cycle wouldn't run. But acetyl-CoA gets made from pyruvate, which was made from glycolysis. Okay? We produce an electron carrier, and we release carbon dioxide, which is one of our first waste products. The other waste product is water. Remember, if we go back, we produce energy, releasing carbon dioxide and water, and I just showed you where these two products are. What you need to know about the TCA cycle is that its function is to release waste products in the form of carbon dioxide and to produce electron carriers in the form of NADH and FADH2. These electron carriers will be used for the electron transport chain in order to make more ATP. So, some things to focus on. Alright. Generally, you're going to need to know some important things about specific reactions that are going on, the TCA and the pre-TCA and glycolysis, and why they occur. But you won't have to remember any of those. You'll just have to recognize the reactions. What you really need to know is, what's the point of cellular respiration? What's the point? What's the purpose? The purpose is to make ATP using energy from electrons. And what goes in? What kind of things go into cellular respiration? Think about that. We got glucose going in, we have ADP going in, we have NAD plus going in. All right, what are our waste products? If we go back to the reaction of cellular respiration, what are our waste products? Water and carbon dioxide. And where do they come from? Try to be able to figure out where those waste products come from. If you think about it right now, where does water come from? If you're thinking glycolysis, you're right. Where does carbon dioxide come from? If you're thinking pre-TCA and TCA, then you're right. So what do we make that we can use? All right. We can make ATP, which can be used by the cell to do everything. And why are specific reactions in glycolysis important? So in general, I'm going to send you a worksheet tonight. You can work on that in order to gain the best understanding of what's going on right now as possible. And before anything else, it's just important to focus on the big picture. Focus on what is going on, why is it going on, where is it happening, and then everything else will fall into place. I'll see you on Monday, and you can come in and ask me any questions on the extra help time before school starts on Monday or Tuesday. We will have a test on Friday or Monday or Tuesday of next week. So it's a good time to start getting serious about this stuff. All right, thanks.